everybody. Thank you for coming. It's a great day of celebration in the Tansy Lab today because today I become a Gator mama, hopefully, for the first time. So Jake is the first student, hopefully, that will come out of our lab today. And these are my disclosures. I've been to this rodeo before. We've had several people graduate and study the neuroinflammation in PD and AD models. Then at Emory, we had the opportunity to study peripheral immunity and the gut-brain axis in neurodegeneration in a whole different set of models and diseases. And at UF, Jake will be the first of several to do work on the central peripheral crosstalk that has become so important in terms of neurodegeneration, risk, and pathophysiology. And as a member of the Tansy Lab 3.0, he has had a good deal of firsts. He was the first to recognize that the DB8 mice stopped expressing human synuclein. That was a sad day in the lab, and he had to veer in a different direction. Then he also was the first to recognize that the DSS colitis model doesn't travel well at all. And the results we were getting at Emory, we weren't getting here. And that led him to optimize a protocol for the colonies that we have here, and you'll hear about that today. He was also the first to use light shade microscopy to image the locus coeruleus, and the first to need hypermeter space for image storage and processing. And finally, he was the first to venture into microfluidics-free single-cell RNA-seq, that I hope he'll tell you about today. So he's shown a fair amount of adventurous spirit and resilience, and I think that's important in grad school because it really does prepare you. He was a closer on this paper that we wrote through the pandemic, and he was critical in doing the immunohistochemistry that we needed to do to show expression. So this is all before the colony, the vax, stopped expressing. And so it's been a hard lesson for all of us, but buyer beware. He's also been, even before his quals at the top there, he was the team player. We do a lot of assembly line takedowns, and Jake is always there and leading the pack and helping out. He's also a great mentor to a number of people in the lab, including Abby Anderson, along with Drew, and Maeve Kruger, who you'll hear about in a minute. He went to a number of conferences while he was here, and he represented us well. Most recently, he spoke at SANCON, and that was a really great opportunity to just kind of show and showcase what he's done here. This is submitting his first paper on bioarchive and brain behavior and immunity. That was a great moment at my house during the lab party. And he's got 2 papers right now as primary author under revision at Brain Behavior and Immunity, and 1 at Nature Com. And you're going to hear hopefully a little bit about this and the tour de force that they did to optimize this model. Maeve Kruger is a master's student in the lab, and Jake has been his direct mentor at the bench. And so, in addition, he did an incredible tour de force paper with Oyane with single-cell PIP-seq, which is what they call, RNA-seq for the masses. So it's a lot cheaper and it's a lot higher throughput, and they got some really interesting findings about the central peripheral crosstalk during peripheral inflammation. And then he's got a 4th paper in process on the locus coeruleus modulating inflammatory stress, and he'll be a co-author on the metagenomic analysis of gut microbiomes in IBD and PD subjects with me. So in addition to all the scholarship, he's had many great relationships, and he's been to Keystone. We've had lab meetings. He's been a great friend with other people in other labs. And finally, you don't make a scientist without a village. And so I want to thank Jake's Scientific Village, starting with his thesis committee, who have given a lot of guidance and a lot of their time. The Movement Disorders T32, who selected him, and I think he's been a great leader there. finally the neuroscience department, and then the institutes and centers that have supported his work and have cheered him on this whole time. So we wanted to go get it and pay it forward. He's going to be going to a postdoc at Northwestern in David Gates' lab, and we can't wait to see the papers that come out of there. So, Jake.
You guys can hear me okay? Yes. Microphone working? Okay, very good. Thank you, Malou, for that very kind and funny introduction with all those really great pictures that we take so often. So I'm going to be defending my dissertation today, and sort of some of the work I'm going to talk about in the lab focuses on three sort of main components you'll find here in the title. Gut, norepinephrine, and brain inflammation. And a lot of the data I'll show you come from sort of mouse modeling efforts based on observations that we and others have made about Parkinson's disease. So these are all the features summarized very quickly here for you. So just to show you kind of what we're going to talk about today, we're going to sketch it out real quickly, and we'll return to this sort of outline periodically just to kind of reorient ourselves as we go. I mentioned that the projects we've done we think are most relevant to Parkinson's disease, so we're going to start with some sort of obligatory intro about what that even is. We'll move into a focus on the gastrointestinal systems in Parkinson's disease, focusing on the dysfunction and inflammation, which will lead us into trying to model that dysfunction and inflammation in a laboratory, which, as Malou sort of mentioned, reveals a whole bunch of problems that would prevent us from moving forward. And this will take us into our first sort of bit of data, where we are looking at how the dysfunction of the gut actually drives inflammation in the brain. We'll walk back out for a moment and talk about Parkinson's disease again very quickly, focusing on the locus coeruleus, which is the brain's main source of norepinephrine, as a possible sort of mediator in the disease itself. And then this will take us into our next bit of data, where we're trying to understand the interaction between gut and brain in sort of a different way, focusing on gut inflammation and locus coeruleus injury. And then we'll be done. So, like I said, we'll start with Parkinson's disease. This is the most common neurodegenerative movement disorder, characterized in the clinic by several sort of canonical features, including rigidity, resting tremor, postural instability, slowness of movement. And generally in the field, we see images like this get passed around a lot, this sort of cartoon of the shaking man more or less. So this is kind of our stereotyped idea of what a Parkinson's disease patient may look like. As I mentioned, it's a very prevalent disease. In the United States alone, generally there's somewhere in the realm of 90,000 new diagnoses each year, and we spend upwards of $50 billion in healthcare costs. So it's quite important that we try to understand what's going on here. In the brain, there's sort of two key histopathological features, which is the loss of the dopaminergic neurons in the substantia nigra. So you can see here's a healthy substantia nigra with a thick black band of neurons that are making dopamine. These guys are generally degenerated in the Parkinson's disease brain. And we also know that there's the inclusion of alpha-synuclein into Lewy bodies, which are kind of a few shown down here. I'm not going to spend almost any time talking about alpha-synuclein, but I'll get yelled at later if I don't at least mention this. So there you have it. What makes this disease really difficult for us to sort of deal with is that over 90% of cases occur without sort of a known cause. We call these idiopathic cases, and these are the vast majority of Parkinson's disease cases are idiopathic. And the other thing that sort of complicates it a little bit for us is in the clinic and in the brain, these features that I've summarized over here generally vary quite a bit person to person. So even though, for example, we have this very stereotyped image of a Parkinson's disease patient, we know that the disease can look like a lot of different things. We know it can affect people to different degrees and affect their lifestyles and progress in different ways and things like that. So it's a little bit hard for us to hone in on certain things. The other thing to realize is that generally we think by the time Parkinson's disease sets on in the clinic, it's too late for some sort of disease staging intervention. So here's sort of a nice schematic I like to think about. And this is showing, sort of summarizing features of Parkinson's disease over time. And you see this big, thick blue band is sort of an estimate of the number of dopaminergic neurons in the substantia nigra. And by the time of diagnosis, here in this dotted line, it's estimated that somewhere between 50 and 80% of those neurons are already gone. There's very little at that point we can do to get them back. So it helps us to kind of look ahead, look forward, and look into earlier symptoms that may emerge that we can sort of think about as predictive in some way. Many of these earlier symptoms are summarized here. Don't worry about it just yet. I'm going to focus on a few. And what's sort of important for us is to realize that these earlier symptoms may be coming from an actual sort of disease process, that it's not collateral damage of some kind. They're maybe actually playing a role in the disease. And you'll notice that one of the very first symptoms that we find years or even decades prior to the clinical onset of Parkinsonism is gut dysfunction. So now we're going to focus on the gut here for a few minutes. If you get nothing else from this next section, I just want you to take away that gut dysfunction and inflammation are very early processes in Parkinson's disease, and we think that they are central disease processes in Parkinson's disease. So this is a nice figure that kind of summarizes all of several different either gastrointestinal comorbidities or features of Parkinson's disease. You can see a lot pertaining to the bacteria in the gut, as well as a few symptoms, including diarrhea and constipation, are very, very common. But the link I want to focus on is this link between inflammatory bowel disease and Parkinson's disease. And so there's a few key things that I think we all need to keep in mind, is that with an inflammatory bowel disease, your risk of Parkinsonism later in life goes up by about a quarter. But if we treat the inflammatory bowel disease with some sort of anti-inflammatory medication, especially tumor necrosis factor therapy, we can shut that risk down by about 80%. And there's an interesting sort of genetic overlap between Parkinson's disease and inflammatory bowel disease, and many of these sort of risk-carrying variants land on genes we know to be involved in the immune system. So we think here that the link between gut and brain, at least, is partially mediated by the immune system, and this is where we're going to spend a lot of time today. So irrespective of sort of clinical inflammatory bowel disease, there are several other features of the Parkinson's disease gut I think are important to remember. Again, I'm not going to spend too much time on it here today, but we do find alpha-synuclein pathology in the intestine, and this seems to be relatively unique, depending on who you talk to the Parkinson's disease gut. And we also find an eroded sort of intestinal epithelium in Parkinson's disease. So this is kind of a very cool strategy that these guys did back in 2017, where you take a colonic biopsy from people, you can stain it and image it in 3 dimensions, and we find that in the healthy control gut, the presence of these tight junction proteins between epithelial cells, and I'll spend more time talking about these later, are generally intact in the gut, and these are helping sort of regulate the paracellular permeability in the intestine. But in the Parkinson's disease gut, you find sort of a loss of these tight junctions. And so the evidence I've kind of summarized here lead us to this kind of gut-to-brain model of Parkinson's disease. We think, I'll summarize this for you, that there's some sort of environmental inflammatory trigger, or maybe several triggers, that land in the gut and sort of kickstart intestinal inflammation and maybe erode that epithelium. And this, in turn, locally may drive the synucleinopathy I'm showing here, which may spread up into the brain. Again, just so you guys know, I'm not going to talk about it probably any more than that. But the consequence we're really interested in is that if this inflammation is of the right kind and maybe severe enough, it will spread out into the rest of the body, maybe up into the brain. And we think that over the long term, this may be injurious to those neurons. And so all of this together puts us at a point where we need some way to study the gut-brain axis in the laboratory. We need some way to intervene and really try to figure out what the different features of this system are that are predisposing the brain to injury. So let me tell you how we can do this. Our sort of go-to tool here, as well as many other labs, focus on use this model called the dextrans sulfate sodium model. This is abbreviated DSS. That's probably how I'll call it for the rest of the day. So this is sort of a cartoon, a very simplified cartoon of what the intestine looks like. You see these sort of like upside down Pac-Man ghosts. These are your intestinal epithelial cells, and between them live those tight junctions. And normally in a healthy state, this sort of layer is separating the bacteria that live in your gut to the immune cells that live in your gut. But when we give mice DSS, which we can do so very easily by putting it in their water, The DSS will travel into the intestinal system where we think it interacts with medium chain fatty acids and calcium, probably of a nutritional source in some way. And somehow, we're not totally sure how this works, but these molecules seem to interact and effectively punch holes in the membranes of these cells, which allows these cells to die. And if they don't outright die, they're injured in some way, and generally we lose those tight junction proteins between them. And this allows the bacteria that live in your gut to encroach on the space normally occupied by the immune cells, which is generally not a good thing. And so your immune system responds very aggressively. Not your immune system, the mice's immune system. And so this DSS-induced colitis is a very rapid and severe form of colitis, generally innate immunity driven. And this is generally how it works. Okay, so this is a very popular model, probably because it's so easy. And we've learned a lot about the gut-brain axis in this model. So generally the consensus seems to be that DSS-induced colitis drives brain inflammation. Okay? But the issue is that we learned and spent the greater part of 3 years dealing with is that this immune response in the brain is really ill-defined. In my case, I couldn't even find it for most of the time. So we realized that there's several problems that we kind of have to address. So number one, everybody seems to disagree about what cells are driving this brain immune response. It's really not clear what's between the gut and brain, and this is not clear if the circulatory system is important or if the lymphatic system is important. And one of the most frustrating things is that studies that claim to protect the brain also generally make the colitis better. So it's very hard to kind of disentangle what the features are that are important. There are also many sort of documented threats to this model, kind of like Malou alluded to earlier, that make it not very reproducible. One of which is generally you would design your experiment and treat your mice accordingly in pursuit of whatever you want to see, which means that you might go about your experiment very different than I would. It's known that the time at which you would get your DSS and the supplier of your DSS affects how well it's going to work. And we think that the tissue that you take and the questions that you ask on the back end of your experiment is really going to influence your interpretation. So we know that different pieces of the colon don't respond the same, and we're not sure at this point if different brain regions are differentially vulnerable to this model. And finally, if I'm telling you that the microbiome is sort of the inflammatory trigger, anything that's going to affect the microbiome is probably going to affect the colitis. So even where the mice are kept and raised is probably going to play a huge effect. And so this will take us into us trying to sort of break this model wide open and solve at least some of these problems so we can make progress at some point. And this will take us to our first sort of bit of data here. So the objective of the study that we put together is twofold. Number one, we want to really critically evaluate the reproducibility of the model, focusing on different parts of the intestine, and we're also going to compare sort of our response to that of many other publicly available data sets and see how well these responses carry over between sites. And this is all, you can find it in our preprint. We spend a lot of paper real estate talking about this. I'm not going to go into it too much today because the thing we really want to focus on is what is happening in the brain. And so we want to understand what kind of immune process or immune processes we're really talking about when we talk about colitis-induced neuroinflammation. And we want to know what the kinetics looks like. We want to know how does it look over time. And really, really critically, I'm getting ahead of myself. So we want to understand how different brain regions respond, and if that's going to be a confound for your experiment. And really, really critically, we want to understand which pieces of the model are going to be sort of best informative of what happens in the brain. Can we make any sort of conclusions about the gut talking to the brain, and if so, how can we do that? And so this is our approach. It's not a very elegant approach. It's the everything all at once approach. But this was done with a lot of help from these 5 ladies up here. Maven Moyani helped me sort of design this experiment and organize this sort of controlled chaos. Cassie and Jenny were really critical in the day-to-day sort of checking on these mice and taking them down, so I'm very appreciative of them for that. But generally, this was our strategy here. So we took some regular old mice and put them on a relatively high dose of DSS for a week, and then let them recover for 2 weeks. And along the way, these green arrows, we took different subsets of mice down, making sure to sort of balance as best we could for biological sex, giving us 7 different treatment groups over the course of this disease, which is marked by sort of a peak disease phase and a long recovery phase. We took the colon, we took the proximal colon, which is said to be sort of more resilient in this model, and we took the distal colon, which is said to be more vulnerable. We took the blood to assay sort of circulating inflammatory markers in the plasma to see what the body's response is. And of course, we took the brain. So we took the cortex and we took the hippocampus, generally because this is what a lot of studies will take anyways, and we wanted to sort of have a direct comparison between our data and others. And if we're talking about Parkinson's disease, we wanted to take the ventral midbrain, which is roughly where the substantia nigra lives, and the striatum, which is where the substantia nigra innervates. So I want to characterize this model for you, and as we go, you'll kind of begin to see where some of the frustration begins. So I'm going to show you what the gut injury in this model looks like, and maybe was a huge part in this right here. And so I told you before that the erosion of those tight junctions that are causing bacteria to get in, are gone in the model, and we can measure that quite easily with a Western blot. So here we're measuring the protein occludin. It's a very commonly looked at model. And before I tell you what the interpretation is, I need to tell you how we do our statistics. So when we do these analyses, you see we have 7 different groups, and we're making pairwise comparisons across all of them. And in order to represent the significant post-hoc tests we use in this lettering system, where groups that share a letter are not statistically different, whereas groups that do share a letter, it's the other way around. Groups that do not share a letter are statistically significantly different, and groups that do share a letter are not different. So this one is pretty easy. The untreated group gets the letter B, and the rest of them get the letter A. That means each of these groups is statistically different from this group. But they are not different from each other. And what we can take this to mean is that with DSS and any amount of recovery, we get a huge sort of loss of the tight junction protein occludin in the gut. We can also measure this at the transcript level, and we find a very similar pattern. The occludin mRNA is also severely hindered in this model, probably reflecting overt sort of epithelial cell death. We can also measure sort of immune activation, generally by gene expression. We can measure the expression of inflammasome or inflammasome subunits, different sort of inflammatory systems. We find sort of a steady and persistent activation of the immune response in the gut. We can measure the expression of particular cytokines, which tells us the same thing. And we can also look at sort of genetic markers for immune cells if you want to take this one. In this case, we're looking at the gene for CD45, and this is also persistently and increasingly activated throughout the paradigm. So we get a very persistent injury response in the gut. But when we look into the circulation, we get signs that this sort of body-wide dysfunction, body-wide inflammation is very transient. And so what I'm going to show you, Maeve did little pieces of this, Hannah Staley, another grad student in the lab, did pieces of this, and I also did a few pieces of this. So we can measure pretty easily a lot of circulating markers with plate-based assays. In this case, we can measure lipopolysaccharide binding protein, which is a protein that immune cells make when they sense bacterial endotoxin. It's part of a normal bacterial response. And this marker in the blood peaks at a very defined time point. It peaks at the 7 days plus 2 days of recovery. It's very interesting, again, very transient response. We can measure C-reactive protein, which is a protein generally your liver makes. It's sort of a broad, vague marker of inflammation. This also peaks at the same time, very discreetly. Circulating lipocalin-2, which is a neutrophilic protein evolved in bacterial control. This also peaks at a very defined time point. And circulating inflammatory cytokines like tumor necrosis factor, KC-PRO, which you may see called CXCL1, and interleukin-6, all share the same sort of very quick peak and very quick recovery. So we have a very persistent gut injury, but a very defined, very limited systemic response. I thought this was pretty interesting. And it doesn't quite make sense when you think about some of it. But really the whole point was to understand what's happening in the brain, right? So in order to do this, we wanted to go about this as unbiased as we could. Okay, so I'll tell you about our strategy. So I told you we treated these mice, we took these 4 brain regions, and then we isolated the RNA, which ends up being 400-something samples of RNA to isolate. Maeve did a huge portion of this. Cassie helped out quite a bit. I do not envy them at all doing this. But we can take this RNA and send it off. In this case, we sent it off to a company called LC Sciences. They're based out in Texas for a very standard RNA sequencing workflow, which looks like this very quickly. Basically, you can fragment your RNA into sort of pieces of a known size, convert them into complementary cDNA. You can stick some adapters on there to help with the sequencing chemistry. And then you can actually perform the sequencing, which involves planting those sort of fragments on kind of like a lawn. You can amplify those those fragments, and as you grow those fragments, you can feed the reaction fluorescently labeled nucleotides, which the machine will count, and this will give you kind of an estimated sequence of what the genes are that you're seeing. We can take those sequences, map them to a reference genome, and count them to get sort of expression levels. There's a lot of very standard downstream analyses that we did in the paper, so please go check those out if you're interested. But I'm going to focus on sort of our most unbiased tool, which is the Weighted Gene Correlation Network Analysis, abbreviated WGCNA. The Weighted Gene Correlation Network Analysis, if you get nothing else from the next few clicks I'm about to show you, is basically a way to put genes that move similarly to one another together and see what they're doing. But effectively, this is a very established framework. It's a very powerful framework. And basically, all we're going to do at first is correlate every gene with every other gene to get what we call a co-expression network. We can cluster those genes based on their co-expression patterns using any one of your favorite clustering strategies. In this case, we used hierarchical clustering, which gives us sort of data like this, where every vertical line is a gene, and they're all clustered into these sort of arbitrarily named color modules. So you can see a big old blue module of genes here. You can see a little discrete red module over here. These are all genes that seem to be moving together. And then in order to identify gene modules that we think are interesting and that we should explore a little bit more, we can relate them to external information, such as traits that we've measured in our own mice, or we can infer their biological function. And we did a little bit of both here. So the first thing I'll show you is a big old correlation matrix of all of our gene modules here on the y-axis, and a lot of these sort of external traits, some of which I've showed you before, here on the x-axis. Here we have one matrix for every brain region we've looked at. The orange here is positively correlated, and the blue is negatively correlated. And a few things should jump out at you. You get 3 modules that are positively correlated with those traits, meaning they're going to be upregulated during DSS. And we get one that's negatively correlated, meaning it should be downregulated. And when we look into those 3 upregulated modules, we get sort of your expected expression pattern, a very transient sort of uptick in the expression of these modules. It does not seem to differ across brain regions. And when we go to try to infer the biological sort of function of this module, it tells us what the literature has always been promising, which is the activation of some sort of immune response. So you can see a lot of different gene systems, gene sets that we think are involved in the immune response annotated here. And we also get some sign that there's some sort of oxidative stress or cell stress happening more generally. So again, this was good. We finally found our neuroinflammation after too many years of looking. And so here we have it. And I told you that there's one downregulated module, and that was the midnight blue module. And we get this sort of expected expression pattern, sort of down during peak disease, and then we get a recovery. And when we try to infer this module's function, we get a lot of pathways that seem to do with myelination. Oh wow, that's pretty wacky. I don't think it was ever shown up until this point that a leaky gut is going to directly cause myelination in a healthy mouse. Until as I was sifting through the data, these guys came out right and beat me and published at the protein level that with DSS-induced colitis, you get a loss of all of these sort of myelin markers in the brain. So they beat us to it, but it's sort of a testament that this effect may be real, which is great. So let me summarize up here real quick for you. Number 1, we find that DSS-induced colitis does indeed drive a brain immune activation. We also see that maybe the colitis in some way stunts the production of myelin or oligodendrocyte function in some way. We're not really sure what the functional implications of that are just yet. But the big thing is that the brain immune activation is very transient, and it's not regionally specific. It's sort of a global, not specific response. So even though the integrity of the barrier looks like this over time, a very sort of quick loss, and it's a sustained loss in the barrier's function, and the gut inflammation comes high and stays high, the systemic immune response is very quick, very short, as is the sort of brain's response. So this kind of tells us why we never found this, because it's such a short window to look for. So we think we've characterized this model pretty well at this point, and at the very least can move forward for our own purposes. But one of the biggest things we kind of realize is, because of the quickness of the brain sort of differential expression, it's really unlikely that this injury alone is going to affect some lasting change in the brain. And a lot of you are going, yeah, obviously. And it's like, okay, yeah, I got it. So we can come back to the sort of prodrome of Parkinson's disease to look for other hints that for things that may be mediating this vulnerability. Right? So that's all of the heavy-duty gut work. We're going to go back to the brain, finally, here in the Department of Neuroscience, and think about the locus coeruleus. So again, from this piece, if you get nothing else of the data that I show you, just keep in mind that the dysfunction and the injury in the locus coeruleus seems to be very important in the process of Parkinson's disease. Okay, so if I show you this sort of schematic again, you'll find lots of other symptoms that emerge early on in the disease, including sleep dysfunction, mood changes, especially depression, anxiety, sort of daytime fatigue, chronic pain. These all come on early, generally before the onset of Parkinson's. And we think that a lot of these symptoms are at least partially attributable to norepinephrine. And we also know that the locus coeruleus, which is the brain's main source of norepinephrine, dies early on in Parkinson's disease. So just some quick neuroanatomy. This is where your locus coeruleus is. It lives in the hindbrain, sort of very close to the fourth ventricle. It's a very tiny nucleus, and it is sending norepinephrine all over the brain, including up through the cortex, through your limbic areas, through the midbrain, locally in the hindbrain, back into the cerebellum, down into the spinal cord. And so you think, wow, those are a lot of symptoms that maybe have to do with norepinephrine. That's because norepinephrine does everything, right? So we know in the Parkinson's disease brain, the locus coeruleus degenerates. This is a recent paper using sort of a multimodal MRI strategy showing a decrease in sort of the number of neurons, or estimated number of neurons in the locus coeruleus, as well as the overall tissue volume in the Parkinson's disease brain relative to healthy controls. And we can also look for those messages the locus coeruleus is sending using PET imaging, using a radioligand that binds to a transporter that lives on these axons, and you can see a healthy control sort of locus coeruleus innervation all over the brain, and it's a little bit sort of stunted in the Parkinson's disease brains here, which tells us that there is sort of neuronal loss, neuronal injury in some way, and my obligatory, there's also synuclein in the locus coeruleus. So, just so you know. We know a lot now at this point about the locus coeruleus, and we know that norepinephrine and the nucleus itself are very important in many different processes in the brain. So, for example, norepinephrine is itself an antioxidant molecule. It's pretty important. And we know that neurons and glia alike express a lot of different adrenergic receptors that upon activation by norepinephrine, very good for the cell. So in neurons, generally we see an induction of sort of trophic factors, sort of health-promoting molecules in the neuron. But in glia, we see that norepinephrine seems to bias an immune cell towards an anti-inflammatory state. So sort of a more protective, more healthy, more everything's okay kind of state. But when we disrupt the locus coeruleus in this review, these two guys here focus on synuclein and genetic defects, and we lose norepinephrine. That's one source of antioxidants that's gone from the brain. We also seem to bias neurons to being a little bit more vulnerable in some way. It also seems that glia tend towards more of a pro-inflammatory state also. And we've seen a lot of these things come out in our DSS model, which leads us kind of to our next hypothesis here that in the context of gut inflammation, the locus coeruleus is probably actually protective of the brain. And so this would take us into our next sort of effort here. So we want to use our DSS model and somehow injure the locus coeruleus and see how these two things interact to derive brain health. And we can do so with a drug called DSP-4. And the DSP-4 is a noradrenergic specific neurotoxin. It readily crosses the blood-brain barrier, which means you can administer it peripherally very easily. And when you do so, it seems to be, at least according to the papers you read, selective for the locus coeruleus. So this sort of model I showed you before has really sort of been validated with a lot of work using DSP-4. We get a lot of these expected effects of inflammation, and oxidative stress, and all that stuff all back out when we use this drug instead of some sort of transgenic line in a mouse. We've never used it, so it's important to make sure your models work before you want to use them. And we find that after a single shot of DSP-4 and you give the mice 1 week to wait, we get a very clear loss of norepinephrine in the cortex. And that is 70% loss, which is pretty severe and pretty robust. And we see that dopamine and serotonin in the brain are left alone. But when we look in the gut, we find that maybe this drug is not that selective. And we get sort of a 30% depletion in colonic norepinephrine. So, okay, maybe this is not that good of a tool. But when you wait long enough, we find that the injury to the locus coeruleus in the brain is very stable over time, but if you wait just 4 weeks, the colonic norepinephrine returns to normal. Okay, so maybe this is a workable model. Let me come over here, because I have to show these pictures, right? Because some might say I spent far too much time trying to stain and image the mouse brain in 3 dimensions without sectioning because I'm lazy, and this ends up being way more work. Basically, we can stain a whole mouse brain for the protein norepinephrine transporter, which is going to very easily mark all of the axons coming out of the locus coeruleus. And you can see overall the mouse brain is very bright. That's because, again, the locus coeruleus right here is sending norepinephrine everywhere. But with DSP4, we get an overall sort of shutdown of those axons, especially in the cerebrum, and the brain is overall darker, and that is because all of those terminals are lost. We can take the same approach in the colon, looking for noradrenergic innervation in the colon, standing for different noradrenergic cell markers, including tyrosine hydroxylase and our norepinephrine transporter again. And this is sort of a control mouse. It does not look too different from our DSP4 mouse in either case. We can actually pull numbers out of these images and quantify them a little bit more rigorously. We just haven't been able to do that yet, actually. So overall, it seems like the drug is good. And then we can actually go on and pair it with our favorite colitis model at this point. So in this sort of first pass look, we treated mice with DSP4 or vehicle, gave them 4 weeks to wait, which is now when we know that the gut recovers from this drug, and then we put them on a pretty high dose of DSS. And we see that the disease itself is not affected. So during DSS, these mice get sick, they lose a lot of weight, probably from dehydration, and just from being ill, they don't want to eat as much probably. And we see that this weight loss is not different between saline-treated animals and DSP4-treated animals. And we also know that we can use the spleen weight kind of as a gross marker for the systemic immune response. The spleen gets really big because the cells are just dumping into the spleen. And this sort of spleen enlargement is also not different between saline and DSP4-treated mice. So the disease seems unaffected, which is good. And we can also look into the gut, the actual injury that I showed you before. So again, we can use the length of the colon as sort of a gross marker for injury. The colon gets short when it's under a lot of damage. And we see that this colonic shortening is not affected by DSP4. We see that the loss of tight junctions in the epithelium, here we're looking at Claudin 5, again measured by a Western blot. We see that this loss of gut barrier integrity is not affected by DSP4. And we see that the induction of inflammation, you're sort of using the abundance of NLRP3 in the gut, is also not affected by DSP4. So, okay, we have a model that seems to be specific, and we think we have a good handle on it. So now what we're going to do is we're going to combine our DSP4 model with the sort of regime of colitis that we've created before that we know elicits that peak systemic immune response and that peak neuroimmune response. And just to sort of emphasize the point, we're giving these mice 2 lesions, one in the intestine and one in the locus coeruleus, but we're going to look in the midbrain where the substantial nigra lives. We hope that you can appreciate that this region is quite distal from both of these other areas. So all we should be seeing is what the midbrain is sort of feeling from these 2 injuries. And we can measure the expression of certain cytokines and inflammatory genes with qPCR quite easily. And we see that if we're looking at the expression of interleukin-1 beta, sort of a very often looked for cytokine, very classical cytokine to describe neuroinflammation, we find that only in the mice in this pink group that got both injuries, DSS and DSP4, activate interleukin-1 beta. Mice that got DSS alone here in the green do not. Mice that got DSP4 alone in the orange do not. And I should point out that this is a male-specific effect. The females don't really respond very well. They're here for you to see. The activation of CCL2, also called MCP1, is the protein name, is another protein name for it. This is an important molecule in bringing immune cells into the brain. This is also preferentially activated only in the double lesion group. You don't see an effect here in either of the single lesion groups, which is really pretty remarkable. Because these are the cytokines that everybody says is always up in DSS, and we never see them, and now we see them. So that's pretty great. We can also look for TNF, another very classically looked for molecule. This seems to be upregulated by DSS, not really mediated by DSP4, suggesting that there's some sort of cytokine or cytokines specific systems that we're modulating with the depletion of norepinephrine. So we were motivated by these data and went ahead with an assay that's a little bit higher throughput. which is the nanostring encounter assay. This is quite a simplified workflow from the RNA sequencing workflow I showed you before. Basically, you just take your RNA, here in green, and bind them to these proprietary probes that have a fluorescent sort of unique barcode attached to them. And then you can just count every instance of every barcode, knowing that each unique barcode corresponds to a gene. And that's going to get your expression level of genes here. So we can identify genes that seem to be up or down regulated with DSS alone and treat this as the response we're seeing if it's modulated. And so here is the fold change of these genes on the x-axis of significance. Here we can use this sort of these thresholds to identify genes that we think are differentially regulated by DSS. We can take those upregulated genes, sort of package them into a module, and see what the expression profile looks like in our samples. And we find, very interestingly, that we get this sort of expected upregulation of the gene module by DSS, again here in green, but this response is overall blunted by DSP4. Okay, that's pretty weird. And we can look at those downregulated genes too, and we kind of get a similar thing. So in the green group here, these genes are, as you would expect, downregulated, but the response is again blunted by DSP4. I haven't had a lot of time to think about what this means because I got done with this, I think it was on Thursday. But it's pretty wacky, and I wanted to share it because it doesn't make any sense. So, just to summarize sort of where we're at so far, we think we've kind of had success in developing sort of a novel mouse model with lesions in sites we think are implicated in prodromal Parkinson's disease. And we spent a lot of time making sure that the models are specific and that they work right, and that you're only intervening in systems at the time at which you think you're intervening. And we see that, very sort of excitingly, the injury to the locus coeruleus promotes the expression of cytokines and chemokines, perhaps in a molecule-specific manner, after colitis. But we also see that this same injury blunts the brain's transcriptional response to DSS. And sort of my working idea, if you allow me to pontificate here for a moment, is that a lot of these genes seem to be injury responsive, or probably part of a normal immune response. And maybe it's actually not a good thing that they're not regulated in the way that they should be with this injury. So that's my working idea, but we can debate about that. In progress, we're looking to examine how the blood-brain barrier is faring in this model, as well as the sort of what the status of different immune cells are in the brain, just at a protein level with immunohistochemistry to kind of further our efforts here. So, if you give me just a couple more minutes, I'm going to wrap up and then we can be done. And I want to leave you with a few thoughts. So, generally, when you attend these dissertation defenses, it seems like everything's great and everything's done, right? And everything's a wild success. And I think we're making progress, but there's a lot of things I think that we need to consider to make more progress. So number one, and I apply this this sort of perspective to both the mouse models we use as well as the diseases we're studying, is we really need to understand the cellular source of inflammation. Because that's really going to determine how we can sort of treat diseases or rescue processes in the brain. We, as Malou pointed out at the very beginning, Oyane and I have sort of spent what was actually a pretty fun project trying to develop a single-cell genomics platform that's easy for everybody. It's so easy that even us two dummies can do it all at the bench ourselves. So involving automated brain dissociation, magnetic selection, and a microfluidics-free single-cell genomics platform. Again, the two of us can do kind of a complicated experiment front to back, all at our bench, for relatively cheap, especially considering other technologies. And generally we think as these technologies improve, you're going to be under a lot of pressure to not use them in all of the information that it provides to you. I don't know how much a single qPCR is going to get us, not for much longer, you know. Moving away from that, sort of a feature that we're missing in our mouse models is the chronicity of inflammation. So in inflammatory bowel disease, which I mentioned has a strong link with Parkinson's disease, we get this sort of lifelong inflammation here represented in this kind of dark blue line. And even though it ebbs and flows, right, it peaks and valleys throughout the lifespan, it is present throughout the lifespan. It's not a single week of oh, I'm suffering and then I'm totally fine, you know. And so the inflammation comes and goes and the tissue damage improves. So there's a temporal component that we're missing. And we get the sense that the chronicity of Parkinson's disease is important too. You know, the neuron loss occurs over a long period of time, giving us the idea that there's some sort of constitutive driving factor probably. So we need to incorporate this in our models, and I'll be honest, we tried to, and they just didn't work out for us very well. So, and the other thing is, and this probably didn't take 4.5 years to realize, and it's probably not going to be new for a lot of the Parkinson's disease researchers in here, but it's really going to be important in our animal modeling efforts to include other features of the disease. So if we're trying to understand where Parkinson's disease relevant brain injury is coming from, we've spent a lot of time today thinking about the environment, treating the gut microbiome as sort of a source of problem, or some sort of modeling some kind of trigger that disrupts the epithelium some way, somehow. We think this is important. Another feature that we've added that is a little bit different is we're looking at how the disease may be driving itself, focusing on the locus coeruleus, which may be a sort of early site of injury, and how that may be affecting downstream disease processes. But we need to really be considering aging is a huge component. This kind of gets into the chronicity component that I just mentioned. Genetic risk is, you cannot ignore that. And of course, we need to think about significant pathology, so I don't get yelled at later. And again, I'll tell you, we tried to incorporate a lot of these features, but a lot of them were the model that broke that Malou mentioned and things like that. So we didn't have much success in these areas. But if we really want to move forward with mouse modeling, which is in some ways a necessary evil when we're thinking about human disease, we need to be considering all of these different factors. Right? So that's how I'll leave you. And with that, of course, there's always so many people to thank, especially Malou, my mentor, our lab, especially these folks here in Bold, for the intellectual support, the moral support, and oftentimes the actual hands-on help in the laboratory, which is huge. Our external collaborators, supervisory committee, and of course my family who's here with me today, and our funding. So with that, I'll take any questions.
Any questions? So what do you think would happen if you use your new model in a chronic, repetitive way now that it's been optimized?
Yeah, it's been optimized. So, we'd have to validate that this modulated immune response is real, right? But if it's true that there's more cytokine being created and released in the brain in this dual hit, then that's probably not going to be good for neurons, which we know are sensitive to too much cytokine all the time. That would be, I think, the most reasonable downstream effect. The other thing we need to worry about is if it's a midbrain specific effect, which it may or may not be, because the locus coeruleus does send norepinephrine everywhere, and colitis seems to drive immune activation everywhere. So it will probably be bad for neurons, but I'm not sure about dopamine neurons specifically.
Jonathan and Becky and
Thank you.
You got a cool thing with the myelin.
Yeah.
So what point after DSS was that data?
So this sort of whatever the opposite of a peak is, where it's down the most. It was at kind of this 7 days plus 2 days time point, and this is where that systemic immune response is the worst, and the other disease metrics that I didn't show, that they're also the worst. So it coincides with peak disease.
And it coincides with their extreme loss of body weight. How much body weight do they lose?
Yeah, so it depends. It's really pretty variable, but put it around 15-20% body weight, but they bounce back in a short time.
Do you think that effect could be like a stress response to like metabolic, like a lack of energy and maybe using myelin as like a desperate?
So this other paper here, they talk about some sort of serotonergic dysfunction that there seems to be lost in DSS brain. Their study was in pursuit of trying to understand why depression and anxiety and things like that would come on with inflammatory bowel disease. And so they really look at serotonin and its downstream signaling. I think it's TRAC-A on oligodendrocytes that affects myelin. I think it is. They say it's a sort of a neuronal dysfunction with a downstream consequence on myelin, like an autonomous sort of system. But I think it could be very much an effect of stress. It's just not something we've been able to do. I think it's an idea.
Your last slide you were talking about the bringing together of different aspects of disease, especially age, right? And I'm obviously looking at your data through a biased lens of what I do, and I'm just wondering with IBD patients, has anyone looked at the changes in kind of like systemic inflammation, immune responses with age, so the IBD will get diagnosed when you're younger,
Yeah.
but then like you said, you have it throughout your whole life. I'm just wondering if, I'm obviously looking at this as like a age-applied immune exhaustion, like I've been looking at BD, but has anyone looked at that kind of concept in IBD?
Yeah,
Or is that something that can connect the two?
to my knowledge, I don't know if there's a lot of like aging IBD reports. It's kind of one of the issues is IBD is sort of a hard thing to study in its own way because it's so bad on patients' quality of life that they treat it pretty aggressively, right? And so by the time that they're sort of like aged, they probably have it under control mostly, or they've had a surgery even to rectify it. So I don't think that's been done. And I hope for their sake that they still don't have bad IBD, at this point. I mean, we know that in mice, you give an aged mouse DSS, everything's just worse. The immune response is just way worse. But what that looks like with chronicity, I don't know.
Great presentation, Jake, as usual. I was looking at, or thinking about kind of your two vignettes and the context of one another, and I was wondering if anyone, or you guys, in any model of DSS has shown specific effects on LC, like the LC region itself.
Yeah, we haven't, nobody has, we tried to with the DBH synuclein mouse, thinking that synuclein would be a source of predisposition. Found out there is no synuclein in the brain, at least at this point, so that did not work out for us. To me, it makes sense that there should be some sort of link. If we're thinking about synuclein, or if we're thinking about the locus coeruleus' own sort of physiology, there's a lot of reasons that the LC might be vulnerable to peripheral insults, including its innervation of the blood and also its proximity to the CSF. So anything that's in circulation is probably going to be bad for it. But that's all hypothetical. I think, like I said, we tried because that's, I think, a really super important, critical question, and it just didn't work out.
Yeah, there's evidence that activation of beta-adrenergic receptors actually increases permeability in the gut. How do you think this kind of fits with your hypothesis that the loss of noradrenaline will be, protected?
Yeah, I've seen both. I've seen that too much norepinephrine is bad and too little norepinephrine is bad. And so it may be a fine sort of balance. So in the gut, I'm not as familiar with norepinephrine. It does a lot of the same things in the gut as it does in the brain, right? It's sort of immunomodulatory and all that stuff. I know in the DSP-4 model in the brain, that's been shown to hinder the blood-brain barrier's integrity. So, like I said, to me, there's probably a balance, and where it's, like if you're doing it in vitro and you just give them a ton of norepinephrine, that's probably going to freak them out just as much as in the brain if you were to take it away. So, I think both things could be true. But we tried to avoid all the gut stuff in the first place, like controlling our model, because it would be it'd be really hard to kind of disentangle.
We'll end with our next PhD defense candidate, Anthea.
Thank you. Very great talk. I was very curious about your two-hit model, and when you showed if there's a loss of norepinephrine, it can exacerbate the immune response, and you also told us that norepinephrine normally would have an anti-inflammatory effect. Do you think that's purely because, a loss of norepinephrine, and if you at this point treat with a MET inhibitor with a DSS, would it help?
Yeah, so you're asking, is it a norepinephrine specific effect? Yeah, we thought about that, and how we could prove that it's norepinephrine, because that would be very interesting. Question, is it norepinephrine or one of the locus coeruleus' many co-transmitters? It's a difficult pharmacological endeavor because you also have the colitis to worry about and try not to, try to keep all your pieces in order. But at the same time, if we're thinking about Parkinson's disease, it's not a specific loss of norepinephrine, it's an injury to the locus coeruleus, so everything else has gone to. Am I getting at your question? I may have only just taken a piece of it. Okay. Yeah. It's difficult.